All right. Um, okay, so with that in mind, our next speaker is Nada Amin. She's a professor at Harvard, and she's going to talk about functional programming for COVID-19 drug repurposing. So, if you've ever wanted to use functional programming to really make an impact on the world on like a really relevant problem, this should be a great talk. So take it away, Nada.
Thank you, Tanya. It's great to be here. I'm excited to talk about a real-world application of functional programming, one that can save lives. The project is led by Matt Might and Will Byrd at UAB, and their funding comes from the NIH NCATS Biomedical Data Translator project over there. And there are collaborators at large as well, and I'm one of them. I joined the project in April but I'm a long-time collaborator of Will Byrd before that. And we have some interns that joined us for the summer, some at Harvard and some at IIT Delhi. And it's worth pointing out that the interns from Harvard are only rising sophomores, so you can make a difference with a new background in functional programming as well. Okay, so what is mediKanren, this project that I'm presenting? It's a reasoning platform for biomedical knowledge. And it's a general purpose platform, but it's used specifically to identify drugs that can hit molecular targets. And at the Hugh Precision Medicine Institute at UAB, this has been done for rare diseases, for cancer, and for COVID-19 as well. And in fact, for COVID-19, there is an ongoing clinical trial with the National VA, the Veterans Affairs Hospital. Okay, so what does mediKanren look like? So it has biomedical knowledge, and the knowledge looks like this. You have a subject and a verb and an object, and that constitutes a claim. So for example, imatinib inhibits KIT gene, or estrogen decreases androgens, or you can have imatinib treats stromal cancer, and so forth. And for each one of those triple, you might have some evidence, like it's a claim with supporting evidence. So for example, you can link to PubMed publications and other things. And here's an example where you string these claims together. So you can have imatinib that inhibits KIT gene that causes stromal tumors. And so you know that imatinib treats stromal tumors. That's also part of your knowledge base. But then you might also find that KIT gene causes mast cell activation, which affects asthma. And you might infer that the treats between imatinib and asthma, which is not in your knowledge base. So you can discover new knowledge using these knowledge graphs. And here at the bottom, we have sort of the general pattern for this kind of example. So inhibits causes, and then inhibit causes, and then something else, disease, and then you can infer a treats link. And so what's interesting as well is that because imatinib already treats a disease, then it's already FDA approved, and you can actually go ahead with medical care for new cases as well. Okay, so here's an example for COVID-19, and that is the example that led to the clinical trial. So you have Tempris that activates the virus, the SARS-CoV-2, and androgens increases this Tempris gene, and estrogen decreases androgen. So therefore, as an inference, you can say that estrogen decreases Tempris gene, and so the proposal was to find anti-androgens based on this fact that androgens increase Tempris gene. And that led to looking at the Degerelix drug, which is for prostate cancer actually, and looking at it to treat COVID-19 patients. So one of the challenge is that there are multiple databases that we have to integrate into one knowledge graph, and we might have even many knowledge graphs. But for one database, there are many sources of information. So for example, you can have a protein database, a disease database, a drug database, a gene database. And these might, and some database might have multiple concepts. So going from proteins to genes, for example, or going from proteins to diseases, and so forth. And if we look at one knowledge graph in particular, it has many relations, many possible relations between things. So for example, you might have an ensemble gene that is equivalent to an HGNC gene, and that's just a difference in identifiers. And then you might have an ensemble gene that encodes a uniprot KB protein. And then you might have this protein that is expressed in cell tissues and involved in pathways using the Go pathways gene ontology database. And then you might have this protein that has a phenotype which is symptomatic or has a disease and so forth. So there are a lot of possible relations, and it can be a little overwhelming to explore these graphs, but at the same time there is so much knowledge in them. So we want programmatic ways to explore this knowledge. And if we look at the original example that I had with imatinib, that treats stromal tumors and also asthma, this is a graph that tries to incorporate a little bit more of the knowledge that is around things, and you can see that the branching factors are exploding a little bit, and so in reality, things is more, it's more difficult to find the critical links because there's a lot of knowledge that is also incidental over there. Okay, so just to go through a few issues, two issues with knowledge graphs. So one is that across databases and knowledge graph, you want to be able to identify concepts. So doing synonymization or normalization. For example, for the TMPRSS2 gene, there are actually two, well, there's more than two, but here are two example identifiers for them, and they both represent the TMPRSS2 gene. And furthermore, at the medical level, we have imatinib versus imatinib mesylate versus Gleevec, and they sort of represent the same drug, but there are a few differences. So Gleevec is the brand name for imatinib, for example, and most of the time, these differences don't matter, so you want to treat these as equal, but sometimes they do matter. And the other issue is that not to be overwhelmed with the results. So just to give an example, SEMED-DB has 97 million edges, and it's just one database among 40 in the RTX2 knowledge graph, which I showed previously. And so you can get an explosion of results, and in fact, you can think of it as any concept with sufficiently many links can be sort of related to one another, like sort of the 6 degrees of separation. And so this makes it hard to actually mine the graph for knowledge that is relevant to a problem. And some solutions here are low-level filtering, so making graph queries and then filtering the result to make it more useful. And then higher-level reasoning, which involves not even doing the full query, but guiding the query engine in ways that are useful. And I'll talk a little bit about both of these. You can say that mediKanren today is more of a query engine that aggregates over many knowledge bases, but we're trying to turn it into a higher level reasoning engine as well. Okay, so let's look at this picture again and generalize it a little bit. So if we were to sort of do this as detectives, we would look at it like this. We want to look at a concept one that decreases a concept two that increases the expression of the TMPRSS2 gene. So I want to now show how you do this with a graphical interface, and then how you can do that with code. So this is a graphical interface that a medical researcher might use. And It's based on the idea of having the subject and the object, and the predicates are the verbs, so to speak. So you have concept one, predicate two, concept two. And so the way I'm going to use this is just put temporis 2 here, select one of the identifiers for it, and say increases, and then here I can find that there is a few things, and including androgens. And now I can put the code for androgens over here. And then click on here, and then decreases, and then here I can see there's estrogen, for example. And if I click here, I can see the PubMed URLs of supporting evidence for this link and other properties of the edge. So that's how you would do things in this graphical interface. Now let's look at how you do things programmatically, and then that allows you to enhance the query results as well. Okay, so I'm going to load this file. And here what you have is, this is a low-level interface to do the same thing, well, to do something slightly different, which is we want to look at both positively regulates and negatively regulates edges. And this is a sort of low-level logic programming interface where this equal represents unification. And so we can see that an edge has this shape where you have a subject, object, predicate, and properties. And so by running this method, I'm asking for all the edges that satisfy this predicate and satisfy this concept, which is set to gene query over here. So if I run this, I get the results of subject, predicates, object. And there's a higher level interface that one can use to do the same thing, which is that you specify the concept. So here we have the gene query, and then S can be anything. And then you specify a link, and you say it either positively regulates or negatively regulates. And then you specify the paths. So if you run this, you see that you get 38 edges, and you can look through them and see that you get a similar list. Now, maybe you have more results here because we're doing synonymization as well. And then once we have that, we can look at the androgen result over here and do a query with the androgen. So looking at what negatively regulates the androgens. And again, we can do this, and we get many results. And let's actually look how many results we got. So 540 results, and one of them should be estrogen. Yeah, so we have 2 results here because there are multiple edges that make this claim. And one other thing we could do is look at both these things at the same time. So the positively regulate link and the negatively regulates link. So making a 2-hop query at once, where we have the gene query and then what positively regulates it, and then what negatively regulates that. And if we do this, we see that we get many more results. So we have 1,779 results for the R. And furthermore, I'm not using the full power of these negatively regulates and positively regulates, because you can define things that sort of takes everything that is sort of synonymous with positively regulates, and similarly for negatively regulates. So this query can quickly blow up if we do it naively. Now, a final thing I want to show is that we actually did a general tool for looking at gene budging, which is this idea of like upregulating or downregulating genes. And so we did it for the COVID genes, which were about like the genes related to COVID, which were about 250. And one of them is this temperance gene. So let's just see what this looks like. So we take the gene, we just do this query, and it's doing something. And now I'm going to show you the spreadsheet that this generates. So here we see that this is really helpful for a medical researcher to just sort through the results. So you have the androgen, for example, that stimulates the gene. And then what's interesting is that you have the link to the PubMed publication, but also for some results, we actually have the sentence that was used to extract the knowledge. And this is very useful for medical researchers to quickly decide whether that's a relevant publication or not. So by doing this, we were able to enhance the work of a medical researcher, and one day they were able to sort through 1,300 drug genes predictions that were generated with our tool. And this contextual information that I just showed with the sentences and the PubMed links, and sort of even relying on the PubMed counts, was super helpful to decide what to focus on. And in the in vitro trials, there were maybe a 40% hit rate in terms of the drug and how they affect the virus. And so eventually we want to keep making the tool more efficient with respect to human times. And one example of doing that is automatic ranking, so that we don't have to look at the exponential blow up of results. Okay, so now let's continue with another thing that was done this summer. That's the work of my intern Marissa Zhang, and then a PhD medical student at UAB called Micropattern. So they decided that the motivating problem was to look at ARDS, so acute respiratory distress syndrome, which is what COVID-19 patients die of. And so they look at, they have a set of genes that have been implicated in ARDS, so they get this as an input. And then the question is, are there approved relatively safe drugs that we can repurpose to help people that have ARDS from COVID-19? And the naive thing to do would be to look at what is overexpressed and underexpressed in such patients, and then regulate the overexpressed genes to be downregulated and the underexpressed genes to be upregulated. But doing this might result in an explosion of drugs, so to speak. And what we can do is find which ARD genes regulate other ARD genes to give fewer drugs. And we also want to do this regulation per tissue type. So for example, what is expressed in the lung might be more relevant than what is expressed in the skin. Okay, so how do we do this? Well, this is the first problem we need to solve, having per tissue type the ARDS genes, so genes in general that regulate another set of genes. So even just having this data structure is useful then to do further analysis. And so this data structure involves a query that has many hops and many many sub-claims if we want. And so I'm just going to show the result of this, of running this query, which is actually looking at a gene. So a gene is the input and what pathways it's involved in. So this is getting to M, and what regulates those, and what pathways M regulates. And this is getting to X. And then we're looking at genes, regulated genes that are involved in X. And both cases, we're looking at tissue type, the Cs. Okay, so let's take a look at that. Okay. So the code is actually fairly short. The long part is this list of our genes. And then what's interesting is the query here, which follow the graph that I showed you previously. And then what is done then is putting things into hash tables to make this queryable. And then doing some counts over this, we get a list of ranked association list of regulated genes. So for example, if we look at the lungs, we get this big list. And if we look at the top element, then that's the top regulating gene. And we can see that it regulates so that's the list of genes that it regulates. And then this is only for upregulation, by the way. We can merge upregulation and downregulation, but it depends on the use case. And here, just to look at what this gene actually is. We can see that it's the tumor necrosis factor. So the TNF gene. Okay, so what have we done? We've used functional programming to help in doing drug discovery for new diseases or for cancer or for rare diseases. And this is all done in Racket here, and Racket gives us a big leverage in terms of being able to iterate quickly on the design of the system. And another thing that is used here is this idea of logic programming or relational programming, which is a pure form of logic programming. And this was shown in the low-level query, but it's also useful in terms of just as an idea in the system that you have this graph database and you have a logic language to query it, and you have a general purpose language and general purpose embedded language that is embedding this logic language, so you can do mixed things in terms of queries and queries and sort of reasoning. And another paper that came in to be useful here, this was a paper called The Art of the Propagator by Radul and Sussman at MIT. And Greg Rosenblatt, who implements most of the infrastructure for mediKanren, he had the idea of using propagators to simplify the constraint system in the query engine that you see, the high-level query calculation. So the reason why this works well is because most of the constraints are local constraints between edges and nodes. And so by exploiting the locality, you can make things much faster. And here, the locality is given by the propagators architecture, so the plumbing, so to speak. And that enables that enables something like, in some sense, one thing less to think about. You just rely on the architecture to do the propagation of constraints for you. And another thing I want to mention for higher level reasoning is this book called Building Problem Solvers, and it's about expert systems that use truth maintenance systems, which are reasoning systems that that do sort of the reasoning engine of an expert system. So the idea there is that you can have a set of beliefs and justification for those beliefs, and then you can even know when you've reached a contradiction and handle that. And in some sense, you can explore your world through beliefs and through different sets of beliefs. Okay, so there are three kinds. Well, in the book, there are three kinds of truth maintenance system. There are more of them in the literature, but the three big ones are justification-based truth maintenance system, and that's based on facts and justifications for fact. That's the simplest truth maintenance system. And then logic-based truth maintenance system are augmented with propositional logic. And assumption-based truth maintenance systems reason about all possible words at once. So in the other truth maintenance system, you have one focus of beliefs, of set of beliefs at once. And in the assumption-based truth maintenance system, you can reason about all beliefs at once. And what I want to show is that these truth maintenance systems are useful a useful part of a reasoning engine. So we haven't really used it we have started to use it in mediKanren, but I'm going to show an older system where we've used it to debug metabolic networks. So the general architecture of a truth maintenance system as part of a bigger system is to have domain-specific data and rules, and be able to input that into the TMS, and the TMS is a general facility. So I'm going to show an example of this. And the example is Biohacker, which is this debugger for metabolic network. And so this just gives us an idea of what we can do, and we're trying to make it relevant to mediKanren eventually. So here you have a metabolic network, and the idea is that you have these compounds A, B, C, D, E, F, G, and you have these reactions R1, R2, R3, R4, but you're not really sure of something. So first, maybe R1 is reversible, you're not sure about this. Maybe R2 shouldn't be there, you're not sure about that. And maybe R4 is also the same, you're not sure if whether it should be there or not. And so so now I'm going to show the system to show how we can debug a network based on experiments. Okay. So this system is based on this Building Problem Solver book, which is quite old. It's from the 90s, from 91, and it's all programmed in Lisp, in Common Lisp. But it's a cousin of Racket as well, so we've been able to port these systems to Racket. Okay, so this is loading. So then, just to give an idea of the demo network, how it looks. So this is the network that I showed previously. here, that's how it looks. So we have R1, which has inputs C and G and outputs A and B. And the enzyme necessary for that is E1, and E1 is catalyzed, is created by G1. And so forth for the other reactions. So let's load this network. As you see, this is written in a domain-specific language, and then it's going to call the truth maintenance system behind the scenes. So if we look at the experiments now that we want to run, the first experiment is positive, and we see that it's consistent with the network. The second one is negative, we have no growth, and it's also consistent with the network. The third one is a false positive, so we think the network, the experiment says that we have no growth, but in particular, the model says that we have growth. And the way to, it gives like the minimal thing to do to be able to get a consistency. If we turn off gene G4, then we're consistent with the model. So because here this F is causing E to happen. And then finally, there is a false negative example. So here we see that it's telling us that we can turn on any one of these things, in particular enabling reaction R2, which was our question. And if we look at in more detail at the false negative, we can see that we can now enable the reaction R2 and see that it's now consistent with the experiment. And we can look at all the reactions that are enabled. And we can look at what is reversible. So R1 should be reversible. And we can look at explicit gene expression, so which genes are on. And now we can retract the assumptions that R2 is enabled and see that it's not consistent with the experiment. So that just gives you an idea of how you can have a reasoning engine that allows you to sort of, from a domain-specific language, get to experiments and things like this. So get to the, so debugging your thinking so that your model so that it matches the experiments. Okay, so this is what we showed. So in reality, R1 should be reversed. R2 has no catalyst in the model, but it's enabled in reality, and R4 shouldn't be enabled. And we find all this by debugging the network. Of course, this is a tiny network, and to be fair, the system didn't really quite scale to larger networks, but our collaborator, Jeremy Zucker, actually managed to use the same idea in a real metabolic model. And the trick there was to use integer programming instead of the more general facilities of a truth maintenance system. Okay, so then we looked at belief maintenance system, which augments truth maintenance system with probabilities, or like probabilistic reasoning, and we've implemented actually the first one, the one based on justification-based truth maintenance systems. And it was helpful to be able to rank the results of a query, but it also has its limitation because it's ranking based on provability as opposed to ranking based on probabilities. So probability instead of probabilities. So we're looking at logic truth maintenance systems again and extending it with the probabilistic logic. And our hope then is that we would be able to do counterfactual reasoning, like in the books Causality by Julia Pearl. So the idea is that we want to be able to say, well, ask what if questions. So what if this gene was knocked out, what would happen? And then we can look it up in our, do the reasoning in our system, and then we can create an experiment that says whether that happens or not. So in short, for this interlude on truth-maintenance systems, what's interesting here is that we can do higher level reasoning and higher level probabilistic reasoning that would guide the engine towards interesting solutions, depending, and interesting would be specific to the domain, what it means. So in particular, even if we look at the ARDS gene cell tissue type, there might be many ways to organize the results, and we want to optimize them in some way. Okay, so just to conclude, ideas in the wild. So we've seen a few ideas from books and papers, and also just general functional programming in Racket and Lisp. And my takeaway here is that ideas from research can really sort of have real world impact. And looking at problems that outside the context of CS can be very enriching, and in terms of solutions, it's sort of gratifying to apply CS solutions. Sometimes it might feel like a hammer looking for a problem, looking for a nail, and so this way there is like a way to apply solutions to the real world. And yeah, so I think the main takeaway is that biology needs CS people, like, because there's a wealth of knowledge, there's a wealth of systems, of biological systems that needs to be debugged and understood. And it's been very gratifying to work on this, because like seeing how the medical researchers reacted to our list of spreadsheets for them showed that, like, sometimes with just a little bit of leverage from our computer science education, we can do quite a lot. All right, thank you.